A protoplast is a cell undergoing division without cell wall, without plasma membrane, without nucleus. So this is the answer, without cell wall. To obtain a virus-free healthy plant from a diseased one by tissue culture technique, which part or parts on the diseased plant will be taken? This is the answer of both apical and axillary meristem. We know at apical region, this is the apical region, no virus growth. So it is help in tissue culture. In mung bean, resistance to yellow mosaic virus and powdery mildew were brought about by mutational breeding, biofortification, tissue culture, hybridization, and selection. This answer is mutational breeding. In mutational breeding, we induce through different like chemical and radiation like gamma rays. So this is the example of mutational breeding. Mung bean is the example of mutational breeding. This is the question in animal husbandry. By whose method was a new bred hazardel of sheep formed by using bikinary ewes and merino rams? You know, this is the answer of crossbreeding. Select the incorrect statement. Inbreeding helps in accumulation of superior genes and elimination of undesirable genes. Inbreeding increases homozygosity. Inbreeding is essential to evolve pure lines in any animal. Inbreeding select harmful recessive genes that reduce fertility and productivity. So we know it is also right because after 4 to 5 generation it shows less amount of fertility and productivity. C also correct because inbreeding is essential to evolve pure lines. This shows pure lines. Inbreeding also increases homozygosity. This also increases homozygosity. Inbreeding helps in accumulation of superior genes and elimination of undesirable genes. This is the thing. It did not eliminate undesirable genes. So this is the answer. The answer is A. Homozygous pure line in cattle can be obtained by mating of unrelated individual of same breed, mating of individuals of different breed, mating of individuals of different species, mating of a related individual of same breed. Here is a question: homozygous pure line. Homozygous pure line we always get through breeding with same breed, mating of related individual of same breed. So this is the answer D. We know two types of breed. One is interbreed and inbreed. Another is outbreed. In outbreed, we see here outcross, crossbreeding, and interspecific breeding. In this interspecific breeding, the main thing is it have two different related species. Two different related species means the species is different. So answer is two different related species. That is interspecific hybridization. Among the following edible fishes, which one of a marine fish having rich source of omega-3 fatty acid? So here is the Mistus, Mangur, Mrigal, Mackerel. So the answer is mackerel. As like mackerel, salmon also, herring, this like fishes also show omega-3 fatty acid. Outbreeding is an important strategy of animal husbandry because it in A is useful for overcoming inbreeding depression. This is answer is right first. Inbreeding depression is overcome through this outbreeding. In B, exposes harmful recessive genes that are eliminating by selection. No, this is not because of harmful recessive genes are not seen in outbreeding mostly. C, helps in accumulation of superior genes. This is also correct. Is useful in producing pure lines animals? No. But the accurate answer is useful in overcoming inbreeding depression. It is also correct, but the accurate is inbreeding depression. So another question: plant breeding. A true breeding plant is one that is able to breed on its own, produced due to cross pollination among unrelated plant, near homozygous and producing offspring of its own kind, always homozygous recessive in its genetic constitution. always homozygous recessive in its genetic constitution. This answer is a cell that telophase stage. Single cell protein, an alga which can be employed as a food for human being is Elothrix spirogyra, Polysonia, chlorella. This is the answer, chlorella. As like chlorella, spirulina, these algae are used in single cell protein. First understood what is single cell protein. A cell converted into mass of cells. This mass of cells produce protein. These proteins are used as edible by human and animals. This is called single cell protein. A technique of micropropagation is, what is micropropagation? This micropropagation is seen in protoplast fusion, no. Somatic hybridization, no. Somatic embryogenesis, yes, because it is the method, one type of method of tissue culture. Somatic means the part, if you take a part of plant, this is further tissue culture. You produce a large quantity of plants or large amount of plant. This is known as micropropagation. So this is the answer of somatic embryogenesis. Soma clones are obtained by plant breeding. No, this is the hybridization. No, plant, not through plant breeding. Irradiation, genetic engineering, tissue culture. The answer is tissue culture. Through tissue culture we gain soma clones. Soma means body, clones means as like, no changes in genetic composition. No change in genetic composition. Consider here a plant. Through plant we take this leaf. If you take this leaf, after taking this leaf, what do we, through tissue culture we produce a large amount of plants. After producing large amount of plants, we see here, with large amount of plant we see, this is called same genetic composition, so it is known as soma clones. Which of the following is not an objective of biofortification in crop? Biofortification, it has some objective. One of the objective, improve protein content. This is correct. Improve resistance to disease. This is wrong because it is not mentioned here. Improve vitamin content. Improve micronutrient and mineral content. This is also correct. But so according to question, the answer is which is not an objective. So this B. Match the list 1 with list 2. List 1, protoplast fusion. In list 2 we see first A, protoplast fusion, plant tissue culture, meristem culture and micropropagation. So one by one you see here. Through protoplast fusion we produce tomato cross with tomato and potato, so we produce tomato. So A2, first finding A is 2, so here is the A is 2 and B is 1. Plant tissue culture, totipotency. Explant produce a new plant. This is known as totipotency. Toti means total, potence means power. And in C, meristem culture. This meristem culture is done in a virus free plant. Then micropropagation. This is called soma clones. This is the answer of D. So the overall answer is B. A2, B1, C4, D3. Mutation in plant cell can be induced by kinetin, infrared, gamma rays and zetine. This mutational breeding, this is known as authorized mutational breeding. In mutational breeding you use a radiation, this is named gamma rays. This is the question of animal husbandry. Which of the following is not a step in multiple ovulation embryo transfer technology, MOET? Cow is an administrated hormone having LS-like activity for superior ovulation. LS-like hormone. So here is the not LS. We provide it follicular stimulating hormone. This is FSH. This is FSH. Means we provide FSH, follicular stimulating hormone, not this. So first is answer is wrong. Cow yields about 6 to 8 eggs. This is correct. Cow is fertilized by artificial insemination. Artificial insemination will provide semen to the female. so it produce zygotes. Then after we do transfer this zygote to the surrogate mother. The D question, fertilized, the option D, fertilized eggs are transferred to surrogate mother. So all are correct, only this is wrong, because in case of LH we provide FSH, follicular stimulating hormone. Inbreeding depression is reduced motility. What is inbreeding depression? Reduced motility and immunity due to close inbreeding. Okay. Decreased productivity due to mating of superior male and inferior female. Decrease in body mass progeny due to continued close breeding. Reduced fertility. Yeah, this is reduced in fertility, productivity due to continued in close inbreeding. So this is the answer. Reduced fertility also, productivity also due to close inbreeding. So the answer is D. This is the question. Which of the following is true for golden rice? It is pest resistance with a gene. So answer is, here is the answer. This is the out of question in NCERT. It is a vitamin A enriched with a gene from a daffodil. The answer is, it is a vitamin A enriched. The golden rice is vitamin A enriched with a gene from daffodil. From a daffodil gene or provide and combine to produce a golden rice. This is rich in vitamin A. The another question, a system of rotating crop with a legume or grass pasture to improve soil structure and fertility is called. So